Welcome back. Jane was worried. At age 51, she was healthy, but her older sister had just been diagnosed with breast cancer. She went to see her doctor, said the question, did she have a problem with this breast cancer gene she heard about? Her doctor said, no, her sister was 65, she wasn't young with breast cancer. She had a single tumor in one breast. There was no other first-degree relatives in the family that had breast cancer. You'll recall these criteria for inherited breast cancer. Even so, Jane urged her sister to get a tumor biopsy and get her BRCA gene sequenced, looking for a mutation that Jane might also have. Not surprisingly, Jane's health insurance and her sister's didn't cover this DNA sequencing because it's very unlikely. Look, if I walked in to a clinic and said, I'd like to get my BRCA1 gene tested for $2,000, the entire sequence, they'd take my money to do it, but it's highly unlikely that I have the gene, first of all, because I have no family members who have it, and secondly, it's highly unlikely that I'm getting breast cancer. So health insurance didn't cover it. So the sisters split the cost, and Jane's doctor was right. There were no mutations in the BRCA1 gene. Still, Jane was afraid. Was she at increased risk for breast cancer? Her doctor took a careful history. Jane had some of the risk factors according to epidemiology. For example, Jane had early menarche. She had started cycling at age 12. And you'll recall, women who start cycling earlier have increased risk of breast cancer because they have more years in which they're producing estrogen. That's the supposition. Jane had her first child when she was 34 years old. You'll recall that the later you have first children, the greater the risk of having breast cancer, just because again, when you have children, you're not cycling. Jane was not yet menopausal. She was 51. So she had menopause that was after age 45. And again, there's going to be more estrogen around for more years, so there's more of a possibility for breast cancer. The assumption is a longer exposure to estrogen can lead to cancer. Her doctor wisely plugged all of this information into a computer model, which does exist for this type of situation, and came up with the conclusion that over the next 10 years, Jane had a twofold increased risk for getting breast cancer. What could she do? The answer was prevention. The doctor prescribed a drug called raloxifene for chemo prevention of breast cancer. Some background. In the 1960s, chemists developed anti-estrogens for contraception purposes. Estrogen, you recall, is involved in the ovarian cycle, so perhaps anti-estrogens would stop the cycle and be useful for contraception. One of them was called tamoxifen, which we've seen before in the course. Tamoxifen binds to the estrogen receptor in the ovary. So here is estrogen. Here is the estrogen receptor in the ovary, and as you recall, tamoxifen binds there. And tamoxifen is like a key that inserts into the lock being the estrogen receptor. Tamoxifen inserts, and it's like a key that you break off in the lock. And of course, the lock is no longer going to work. Estrogen, the normal key, can't bind to it, and so presumably the ovarian cycle would stop, and it might be useful as a contraception. But it wasn't. So people said, well, maybe tamoxifen is useful for other things that respond to estrogen, like breast cancer. So since breast cancer is stimulated by estrogen, the question was, can tamoxifen be useful as a breast cancer treatment? The answer? Yes, it is, and it's used now. We talked about this earlier on in the course. It's an estrogen receptor inhibitor, and it's used in breast cancer treatment. Then the question was asked, would it also prevent breast cancer? If the initiation and development of breast cancer also involve estrogens, we might stop it before it really starts. So a clinical trial began on higher risk than normal women like Jane, who had all these risk factors for developing cancer. Called the tamoxifen prevention trial, and the result of the trial was quite good. Here is a graph that shows you the cumulative rate of cancer incidence per 1,000 of invasive breast cancer in women who were taking tamoxifen for up to 6 years. Women who just took a placebo pill that had no tamoxifen but looked like that pill had a certain rate. They developed about 40 cancers. And here it's cut in half by tamoxifen. A terrific result. Reducing cancer, breast cancer in women at increased risk by 50%. A problem. Like many drugs, tamoxifen has and had in that trial side effects. Over 5 years, it might prevent these breast cancers, but typically over 5 years for these 1,000 women, there would be 5 adverse events, like blood clots, endometrial cancer, etc. That's 5 per 1,000. That may be unacceptable, so patients were very reluctant to take the drug as a preventive for cancer, and physicians were reluctant to prescribe it. The solution was the development of a second-generation inhibitor, a molecule like tamoxifen that would insert into that lock, that would insert into the receptor for estrogen, and wouldn't turn it on or stimulate cell division, break the keys, so stopping estrogen, but not having those side effects. And it was raloxifene, the drug that was prescribed to Jane. Of course, we had to compare, as we do in clinical trials, the new drug, raloxifene, to the current drug, tamoxifen. And again, in this instance, first of all, the endpoints were 2. Number 1, what is its effect on cancer? And second, what are its effects on adverse events? So we had 2 endpoints in this clinical trial comparing those 2 drugs. Here is the result for invasive breast cancer. It was called the STAR trial. Gain cumulative incidence per 1,000, and the time in months going up to 6 years again. And there's 2 graphs there. You can't tell them apart. One's tamoxifen, the other's raloxifene in terms of preventing cancer. What about the side effects? The adverse effects, instead of 5 per 1,000, were 2 per 1,000. We're now on 3rd generation of these drugs. The raloxifene is gradually being replaced by a 3rd drug, and that has adverse effects even lower. Jane's doctor prescribed raloxifene to Jane, and she was the exception, this doctor, rather than the rule in prescribing raloxifene as a preventive for cancer in at-risk women. 3 reasons. First of all, there was a worry about side effects. It's quite well known in the media that there's side effects to these drugs. But again, if you notice the balance versus side effects versus benefit, right? Reducing cases by 50% versus a 1 in 1,000 chance of a 1 in 500 chance of an adverse event of some sort. And if it's endometrial cancer, you could treat it. If it's a cardiac thing, it's not killing you, but it certainly is an adverse effect. So the balance seems to favor benefit. Secondly, people aren't used to taking a drug to prevent cancer. They regard cancer as something that you treat. You don't take a drug to prevent it. You might stop doing activities to prevent cancer, like smoking, but taking a drug just doesn't resonate with people. On the other hand, they have no problem in taking cholesterol-lowering drugs for heart disease. The statin drugs are quite famous for this. So they lower the rate of heart disease, and yet people seem to be reluctant to take drugs to prevent cancer. Third, doctors, especially primary care doctors, have no measure, taking these anti-estrogens, no measure to show that it's working. You know, when a doctor gives you a statin drug to prevent cholesterol, to lower the cholesterol level, they can measure blood cholesterol. They give you the statin, and they'll monitor your blood cholesterol. With these molecules like tamoxifen or raloxifene that work inside the cell, there's no way. You really can't measure, you can't show much of a significant change in estrogen levels in blood or with the receptor activity. There's no easy test for it. So the lesson in all of this introduction is there is science to prevent cancer, but some of it's not acceptable yet to the people, either the patients and or their physicians. In the remainder of this lecture, I'm going to talk about cancer prevention. And I'll do it in 2 ways. First, I'll talk about avoiding things that cause cancer. You know one of them is going to be smoking. And secondly, I'll describe how we do these studies like tamoxifen or raloxifene, how we identify things that might prevent cancer. We can identify preventive agents and the challenge of being able to do that. First, avoiding carcinogens. there are a number of targets for cancer prevention. If we look along the multi-step model for cancer, and let's go along this model, we have a number of things like chemicals and radiation and viruses that it can get into cells. Then you get some genetic, of course, these genetic changes lead to an initiated cell that is now a potential cancer cell. And then more mutations happen, and you end up with oncogenes and tumor suppressor genes mutated, and we end up with a bunch of cells, a pre-neoplastic lesion, and then a malignant tumor, and then a tumor that's malignant and spreads. Where prevention is going to be useful is, first of all, let's avoid the things that we know cause cancer. We can't avoid spontaneous cancer. We cannot avoid that because it just happens. It's chemistry. Certainly, we can try to avoid things we know cause cancer. So that's avoidance of the risk. Chemoprevention generally acts on the initiated cell. So it's going to stop that cell that's been mutated just with those first mutations or two, and it's going to stop it from developing into cancer. So those two steps, avoidance and then chemoprevention. So first, avoidance. The causes of cancer from earlier on in the course, the pie chart, the famous pie chart. The two major causes that we can deal with are tobacco and diet. So let's look at them in turn for prevention studies. In any situation where you're asking people to avoid a risk factor, to change their behavior or the diet or whatever they're doing, the person is going to do risk analysis. To review, risk analysis involves identifying the hazard. Does it cause cancer? Dose response. How much cancer is it caused? By what dose? How many cigarettes can I smoke? Whatever. Exposure assessment. How much it gets into you. And finally, risk characterization. Is the exposure and the cancer I could get from it worth the benefits? So if we're asking people to change their diet or change their behavior, people are doing, whether they are aware of it or not, they're doing this risk analysis. And you'll note that in risk analysis, some of these are scientific judgments and some of them are social judgments of costs and benefits. In 1643, a Chinese scholar wrote that 1643, smoking tobacco too much blackens the lungs and leads to death. 1643. The result was the Emperor Weizhong outlawed growing and smoking tobacco. Great law, terrific. That's prevention, right? Outlaw growing and smoking. And in fact, violators were beheaded. Unfortunately, a year after this law was promulgated by the emperor died along with the Ming Dynasty, and not from tobacco, by the way, and tobacco use came back. So now we're going from 1643 to 1988. In the state where I live, in California, we passed a ballot initiative that taxed cigarettes at 5 cents per pack, and a lot of that money goes into prevention. There's a lot of people who live in California and a lot of cigarettes smoked, and it's about $100 million a year goes into prevention studies and prevention campaigns. The result has been, and this is nationwide, a decline in smoking in America. You've seen this graph before, and the decline in smoking began about the 1980s and continues now. That's about 50% to what it used to be on a per-person basis. There's been a lot of social legislation passed to prevent cancer by smoking. Public places, it's very difficult to smoke in a lot of locations in the Western world. Supermarkets, even where I live on the beach, worried about secondhand smoke. Smoking is now an outlaw activity in a lot of our society. The result is going to be a lung cancer decline. It's just beginning, but you'll recall from our studies of epidemiology, when smoking began in earnest, it took about 20 years for people to develop cancer. Same token, smoking stops, it'll take 20 years for cancer to be reduced somewhat. And as we discussed earlier in the course, it's very difficult to quit because nicotine is addictive. So smoking, pretty easy target. Diet, a little more difficult. Epidemiology links diet and cancer, but the question is, what do we avoid? What are we supposed to avoid? I described earlier in the course that there are many carcinogens that are natural. There are these plant pesticides in food. There are also things we think are good for us. So people have done cohort studies where you might put, the ideal would be to put people on a modified diet. Say, eat your fruits and veggies in a group that don't, right? and see if the group on fruits and veggies get less cancer. It's very hard. You know, Jane might have been asking the question in the opening story of this lecture, could she go on a diet to prevent development of breast cancer rather than take the raloxifene that she's taking? It's hard to do intervention studies on people and diet. It's very hard to control what people are eating. Instead, we do these retrospective or following studies where things like the Women's Health Initiative, where there are many, many women, tens of thousands of women, and we look at their diet and we classify them according to diet and then follow them and see if they get cancer. So, for example, 40 centers in the Women's Health Initiative, postmenopausal women, 30,000 have been described on a regular diet, that's normal American diet, and 20,000 on a diet that's lower in fat, and increase in fruits and vegetables. So you've got a rich fruits and veggies, good for you, 20,000 people and a larger group that's not especially eating more fruits and veggies. So they've looked for breast and colorectal cancer development over a period of decades in these women. And the answer is, does a diet rich in fruits and vegetables affect colorectal cancer and breast cancer? And so far the answer is no. So here is the cumulative risk hazard, and here are the years. We're going out to about 10 years here. And there are people, it's a comparison group and the intervention group. It's not really the intervention, it's the people who ate a diet rich in fruits and veggies. And there is no difference so far, fruits and veggies and breast cancer and colorectal cancer. Does this say don't eat a diet rich in veggies and eat a high-fat diet? It doesn't say that at all, because there are many other benefits to the things, to that type of diet. It just may not be for breast and colorectal cancer. Here's another example, the EPIC study. The EPIC study is following the diets of tens of thousands, hundreds of thousands of people in a number of European countries. Here they are. 520,000 people enrolled in the 1990s. We're following them now for over 20 years, looking for diet, nutritional, lifestyle, and cancer. So findings so far for breast cancer, looking at people who eat a diet rich in fruits and veggies versus not, again, no effect on fruits and veggies at preventing breast cancer development. In addition, phytoestrogens that you hear of, which are estrogenic compounds in plants that have been touted as a possible preventive agent, no effect. Obesity does increase cancer. We knew that already. So it's very difficult. Governments are still recommending fruits and veggies to prevent cancer, and in popular books, you'll read many popular books that do it. The data are not showing that there's that effect. Many clinical trials are underway looking at dietary effects, but it's very, very difficult. It's like identifying what in the diet causes cancer. You remember how hard that was to do. So hopefully we'll make some progress over time, but it's going to be slow. Some more obvious ways to prevent cancer that related to diet. For example, sunscreen and melanoma. That's easy, right? UV light and skin cancer. We block ultraviolet light. We can block skin cancer. In Caucasian people with fair skin, an SPF 16 sunblock will prevent 95% of the ultraviolet light from reaching skin cells, and there have been clinical data now and cohort studies showing that that will prevent the development of both lethal and non-lethal skin cancer. Vaccines can prevent cancer. So I mentioned this earlier on in the course. Hepatitis B virus, causing liver cancer, involved in the cause. Human papillomavirus, causing cervical cancer. There are vaccines now against them, and they're now widely used, and they will lower the rate, definitely, of those cancers. We're just beginning to see that. Chemoprevention is the identification and use of non-poisonous nutrients or drugs to prevent cancer. Non-poisonous nutrients or drugs to prevent the development of cancer. And the idea is to enhance the body's own mechanisms against the development of cancer, or to take the cancer cell that's already initiated and stop it from working. The hints that chemoprevention might work very often come from epidemiology. People on some diets and foods get less cancer, so you look for the ingredients. They come from studies of drugs. Sometimes people who are taking certain medications have less cancer, and these drugs might be stopping an activity that's involved in cell division for another reason. For example, anti-inflammatory drugs that are used to treat pain and rheumatoid arthritis seem to also block the same molecule involved in pain and inflammation that signals cancer cells to divide. So it's especially true of colorectal cancer, and aspirin is being shown now to be a preventive for the development of colorectal cancer. Identification of chemopreventive agents is typically done by experiments on model organisms followed by human clinical trials. 3 levels of experimentation. First, cells, see if it prevents cancer in cells. Then animals, see if it prevents cancer in animals. And then we'll go to human studies. So first, cells, screens of cells. There are whole laboratory buildings devoted to this at various cancer institutes around the world. Cells from tissues like lung and ovary are put in a laboratory dish, and there they are. And then you add a known carcinogen, right? And so they get transformed into cancer cells. Okay, that'll happen in the laboratory dish. So there they are, transformed in the graphic. If you add the carcinogen in the presence of our suspected chemopreventive agent, maybe you'll get no transformation. That'll be good, right? The chemopreventive agent at the cell level prevented carcinogenesis from happening. We can also do chemical tests on cells to find out how this chemical works. The second type of study, we can do our animal screen. So we can take rodents and we can, that can develop cancer, and some of them develop them spontaneously. Others, you have to give a carcinogen to develop cancer. And you do the same experiment we did on the cells. So you take a bunch of rodents and you add the carcinogen, they get cancer. You add the carcinogen plus the preventive agent, they get less cancer. As we saw with the same type of experiments and looking for things that cause cancer, chemoprevention studies are going to take about a year in rodents, and they're going to be very, very expensive. The result of these first 2 lines of experiments show that a number of natural substances and synthetic substances seem to be preventing a wide variety of cancers. Here's a partial list of some of them. There's a molecule called curcumin. It's from turmeric that is very good at chemoprevention in these animal and cell studies. Some of the others, psilocybin from milk thistle. I actually do research on that. Catechins from Greek tea. I do research on that as well. Lycopene, you've heard a lot about from tomatoes. Some medications, aspirin I've mentioned already. Theophylline, indomethacin for pain relief. Verapamil, used anti-angina, seems to be a cancer preventive as well. The 8 substances I mentioned are all in various types of clinical trials in cancer prevention. Again, these clinical trials are long-term, so the results of these clinical trials are going to take many years to develop. Here's some examples of some chemoprevention clinical trials that are of interest. Here's some of natural substances. For example, there was a trial on colon cancer in postmenopausal women, and they were given vitamins A and E supplementing in their diet. The result over a long period of time was no change. There's a famous trial of beta-carotene and vitamin A. It's called the CARET, C-A-R-E-T, carotene, and vitamin A retinol, so it's called CARET, as preventive agents. It stands to reason this might prevent cancer. So they gave this to lung cancer, to smokers who are at risk for developing lung cancer. 18,000 men and women aged 50 to 69. They had a cohort group, so they had an experimental group who got the beta-carotene and vitamin A, and the group who didn't. And you would expect that the beta-carotene and vitamin A would have prevented the development of cancer to some extent. It didn't. They got 28% more. 28% of them more got lung cancer. And in fact, those on the vitamin and the beta-carotene, there were 17% more cancer deaths. To put it mildly, the scientists were shocked. The study was stopped, and this shows you what we really don't know. Drugs. I mentioned earlier on in the opening story of the lecture. You can have anti-estrogens, and they will be useful in chemoprevention of development of breast cancer. What about prostate cancer? The prostate, as you saw earlier on in the course, requires the hormone testosterone to develop. There's a drug called finasteride that prevents prostate cancer by blocking this hormone effect. It's very similar to tamoxifen in women. So a study has been done of 18,000 men over the age of 55. Half of them were given placebo control and half of them given finasteride over 7 years. And the endpoint was a prostate cancer, and there was a 25% reduction. Now bear in mind, it's not 25% of all the men. Prostate cancer is going to be fairly rare. But of those who, compared to those developing prostate cancer, 25% reduction in those who got this drug. Interesting. Few doctors these days prescribe finasteride for men above age 55 and 60. So it really points up the need for education of primary physicians in the use of drugs to prevent cancer. So to summarize, I've described some 2 ways to prevent cancer. One way is to avoid carcinogens, things that cause cancer, and the other way is to identify preventive agents. And both methods are being tried. Some of them are easier than others. Identifying carcinogens, of course, is very hard, and identifying agents that prevent cancer is challenging. Let's step back and look to see where we've been in the course. I want to look back and I want to look forward for a couple of minutes. First, the challenge of cancer. 1 person in 3 in the United States and Western cultures gets malignant cancer. 1 in 4 dies of cancer. That's the challenge of cancer. In part 2, I discussed the causes of cancer. And I discussed population observations and the science of epidemiology, looking for the causes. Cancer, as we saw, is mostly a disease of changes in DNA. The major things that cause cancer are diet and tobacco, as well as some reproductive factors. And I made the point that many of the things that cause cancer are natural rather than artificial. Then I dealt with cancer as a disease in the 3rd part of the course. We looked at cells that were normal cells and saw how they become different when they're cancer. They have different characteristics, and looked at all of these changes. And that these changes happen in many steps. It's an accumulation of damage that happens over many, many years, multiple changes. We saw how the new science of stem cells relates to cancer, and that as a tumor grows, and even when it's initiated, there's great heterogeneity in the tumor. All tumor cells are not the same. We found out that science now knows a lot about the steps of metastasis, how that happens, how cancer spreads, and angiogenesis, how a tumor recruits its blood supply. In the 4th part of the course, we peered inside the cell and looked at genes and viruses and molecular biology. We found that 10% of cancer is caused by tumor viruses, and that led to the discovery of oncogenes in viruses and proto-oncogenes in cells. And 10% of cancer is inherited, and that led to the discovery of tumor suppressor genes. We saw that you can do a molecular biography of cancer and look in very great detail of all the individual changes in specific genes that happen as a cancer develops. And this amazing achievement is giving us targets for targeted therapy. We looked at cancer's treatment and the science behind it, surgery, radiation, chemotherapy. We looked at how cancer is spread and how this led to changes in surgery. We looked at new forms of radiation therapy that's based on science, as well as to minimize normal damage. And we looked, of course, at chemotherapy, from natural products to isolated molecules to the new targeted therapy. And finally in the course, we looked at screening and prevention. It's done on a population basis, and we saw how screening depends on the knowledge of cancer, for example, cervical cancer, cell biology, and once we know the causes, we can get at prevention of cancer. Let's look forward. What do I see on the horizon? What science may know about cancer? I'm going to look at this in 4 ways: the causes and molecular biology of cancer, some aspect of treatment, and prevention. First, the causes of cancer. I think that epidemiology, studies like the EPIC study, these long-term studies with a lot of people, are going to reveal the part of the role of nutrition in causing cancer, and hopefully the issues of what nutrients are involved in cancer will get resolved. We're going to need better methods for screening things that cause cancer because we're missing 1/3 of them in the screens, and I think we'll develop that through DNA technology and rapid genotyping technologies. At the molecular level, sequencing whole genomes of people and their tumors is happening at a great pace as I speak. looking at the differences between normal and tumor cells, at the mutations that are the drivers of cancer and those which are the passengers, is going to give very important results. As we characterize gene changes as a tumor develops, we'll get better biographical information of how a tumor develops at the molecular level, and this is going to give us terrific tools in being able to predict what's going to happen when a person is diagnosed and then deal with it accordingly. What do I think is going to happen about treatment? The more science we know, the wider the therapeutic index is going to be and the less damage to normal tissues. Laparoscopic surgery will continue, that is minimally invasive surgery. Most kidney cancer, for example, treatment is now laparoscopic. Hopefully there will be minimal damage due to surgery and radiation with some new techniques. Delivery of drugs to tumors may change through nanotechnology, tiny structures aimed specifically to home in on the tumor with drug in them. Finally, targeted therapy is going to continue. It's a very exciting field looking at these targets, and the opening story of the course, Gleevec on Kareem Abdul-Jabbar, is going to be replicated hopefully with a lot of tumors. Prevention. Tumor markers, circulating cells in tumors may detect cancer very early, as can radiography. Drugs can prevent cancer, so there may be more acceptance. Most dramatic is going to be the reduction in smoking and hopefully the reduction in less cancer, not just lung cancer. Overall, while cancer is a daunting disease, the efforts of science to understand it have borne fruit. And we are on the verge of tremendous progress in reducing the personal toll that this disease exerts on us all. Thank you.